Lesson 5 Biotechnology Principles and its Processes. So, students, in the last class, I discussed about the mechanism of how genetic engineering is done and how a recombinant DNA is obtained from a desired organism. Now, in today's video, I'm going to discuss about the last two steps, that is, about the obtaining the foreign gene product. The foreign gene product, that means the desired DNA from the desired organism from where we want the character in the host organism. So therefore, the insertion of the recombinant DNA into cells having codes for specific protein is done by the different methods that were already discussed together. So now this recombinant cells needs to produce in large scale. So as you know now that this cannot be done in the laboratory. So these are required to be done in large scale so that different industries or pharmaceutical firms, so they can use it and we get the desired gene product. So in order to produce it in a very large scale, so there is a tank that is used, which is called as the bioreactor. So a bioreactor provides the optimal condition for achieving the desired product by providing optimum growth condition. So there in the tank, so this is like a vessel, a very large vessel, and there the culture media that is kept there. The tate jibilak etia process jikhini choli asile mane bioprocess engineering or jikhini tat এতিয়া কাম কৰিব লাগিব মানে প্ৰিন্সিপলছ টু যে তাত আছিলে তো হেখিনি এটা তাত এতিয়া কি কৰিব মাইক্ৰোবছ বিলাক গ্ৰো কৰিব কাৰণে তাত কালচাৰ মিডিয়া বনাব লাগিব আৰু হে কালচাৰ মিডিয়া বিলাক এতিয়া কত থোৱা হ'ব বায়োৰিএক্টৰ বিলাকত নহয় থোৱা হ'ব এণ্ড দিস বায়োৰিএক্টৰ so there this is an instrument where the optimal conditions for the growth of such microorganisms can be provided by regulating the temperature, pH, the substrate, salts, vitamins, oxygen, etc. So in that way, so in a bioreactor, a large volume of culture can be made under controlled condition where the living organisms could grow. Now what is a bioreactor? So it is a large vessel containing appropriate medium or raw material where recombinant cells are added, and the appropriate condition of temperature, moisture, pH, etc. are maintained in the vessel. So this vessel is constantly stirred for aeration. So as you know that aeration that is very much required, and here aerobic, mainly the aerobic bacteria or aerobic conditions, because our eukaryotic cells, so they require the aerobic condition, or many bacteria are there which are aerobic in their respiratory process. So vessels need constant stirring for the purpose of aeration so that adequate amount of oxygen is obtained there. So after a specific interval of time, the cells are removed and new medium is added, and thus the process of production of cell in large quantities continues there in the bioreactor. So there are normally two types of bioreactors, two types of stirred bioreactors, and these are, one is the sparged stirred tank bioreactor, and other one is the simple stirred tank bioreactor. So this stirred tank reactor is usually cylinders or with a carb basis to facilitate the mixing of the reactor content. So as you have seen in the two diagrams that there are some stirrers or fan-like structures. So these fan-like structures, they provides mixing of the oxygen availability there in the bioreactor. So normally these bioreactors, they are made or they are designed in such a way that every condition for the growth of the culture medium can be monitored from or for the culture of the microorganisms, and so that the culture medium do not get wasted. So you can see there in the diagram that acid base pH control or steam sterilization, because sterilization that is also done here. Then foam breaker, so that is another very important part there in the stirrer, that because foams, so they can interrupt the growth of the microorganisms. These blades, they will help in stirring the culture brood or the culture medium, and the sterile air there in the simple stirred tank bioreactor, so that is kept right at the bottom, so that the carbon dioxide rich air could pass by through that outlet. And in case of the modern one, that is the sparged stirred tank bioreactor, so there also such kind of facilities are given where the microbes can be grown easily. And after culturing those microbes and getting the desired proteins or the products, so cells are then purified through a series of processes called as downstreaming processing, and a purified product are then subjected to taste in different levels. So for that also various testing takes place, and after that they are let out of the pharmaceutical firms or the industries for the use of the public, or they are released for the public service. So in that way, after genetic engineering, these two steps that are generally done in industrial level, so that we can obtain the products from genetic engineering. So this is all about today's chapter. Thank you.